Good morning, everyone, and thank you to PPMD for allowing us to chat with you today about Eutrophin modulation, which has the potential as a universal treatment approach in DMD. Now, before I get started, we are a public company, so I do need to show you this slide. It basically says that I cannot tell the future, and if I could, I'd probably be in a different line of work, so we can move beyond that. So we're headquartered over in the UK in a small town outside of Oxford called Abingdon, but as you'll have noticed by now, I do not have a British accent. I am in our Cambridge, Massachusetts office in the US. Our technology actually came out of the University of Oxford, out of the lab of Professor Kay Davis. And Kay has dedicated her life's work to the research of Duchenne muscular dystrophy. And through this work, what she found is that there's this protein that's called Eutrophin that is very, very similar to the protein that the kids are missing in Duchenne called Dystrophin. And what she discovered is that if she could get muscles to make Eutrophin instead of dystrophin, that this could be a good treatment approach for DMD. And so that's where we come in, and we were founded to find drugs that could actually modulate Eutrophin in the muscles of these kids. Now, I know there's been a lot of information thrown at you over the past couple of days, and there will be even more today, so if you forget a lot of what I say, it's fine. But I do want you to remember a couple of things. The first is that Eutrophin modulation has the potential to slow or stop disease progression in all patients with DMD. And that's because it's independent of the dystrophin gene mutation. And also, this could be complementary to all the approaches you've heard about. And in that vein, we actually have a collaboration with Sarepta. What the collaboration actually means is that if a Eutrophin modulator is approved, that they would take care of selling it in Europe. But until we get to that point, we have access to all of their expertise in developing Duchenne drugs. And I think that's really important as we go along. Now, as I mentioned, dystrophin and eutrophin both look similarly and act similarly in muscles. So at the top, you don't need a degree in molecular biology to see that these look very, very similar. Now, eutrophin is a bit shorter, but it has the important business ends that dystrophin has. And so these both act in muscles as these shock absorbers, like springs on a trampoline. But they do so at different times in muscle development. And so when a muscle is first being made, or when it's being repaired, eutrophin is present in the muscle membrane. And then, for whatever reason, we're still not quite sure why, eutrophin turns off and dystrophin comes to take its place in a mature muscle fiber. Now in the DMD fibers, eutrophin's still made, but it's also still turned off. So it's not the presence of dystrophin that turns eutrophin off, it still turns off. So without dystrophin and without eutrophin, the muscles are prone to damage and can waste away. So what we're trying to do is we're trying to keep that eutrophin turned on so that in mature muscles, the muscles have eutrophin there and don't waste away. Now this is showing this actually in some DMD and non-DMD muscle. So on the left-hand side here you see dystrophin, and in the non-DMD muscle you see that the typical honeycomb-like appearance showing the outside of these muscle fibers. In the DMD muscle, of course, that dystrophin is not there. On the right-hand side we look at eutrophin. So in a non-DMD muscle there is a little bit of eutrophin there, but it's not much. But in the DMD muscle, you actually have high amounts of eutrophin present because there's a lot of repair going on. But that image is pretty patchy, so it's only in spots where the muscle is actually repairing. And so what we want to see happen is that if we are able to modulate eutrophin, we want to see eutrophin looking much more like that dystrophin image of the honeycomb-like appearance. So on the left-hand side here, this is an MDX mouse, which is missing dystrophin, and these are two different muscle types. On the left-hand side is what eutrophin looks like in a DMD case, so that we have that patchy appearance of eutrophin. On the right-hand side is when we give that mouse that's missing dystrophin our eutrophin modulator that's called Ezutromid. And so we start to see that honeycomb-like appearance. And this, to us, and we've also looked at functional measures, but it shows that eutrophin can go to the places that it needs to go to substitute for dystrophin. And so we'll be looking for this in our current clinical trial called Phaseout DMD in the muscle biopsies. Now, before I get to our clinical trial, I do want to thank all of the charities and patient organizations that have been involved with us so far, and we're certainly going to be partnering with them in the future as well. So Ezutromid is orally administered. It's a liquid, and right now in the trial, the kids are taking this twice a day. So far, it's been well tolerated in phase 1 trials. So we've had that in about 100 healthy volunteers and 22 individual DMD patients. So we are now in our phase 2 trial that's called Phaseout DMD. Now enrollment is complete, so I won't really go over the inclusion and exclusion criteria, but I really want to chat about what we're looking for in these trials. And importantly, we expect to report the first results from this trial, from the halfway point in the trial, in the first quarter of next year. Now in our clinical trials, we've been asking very important questions so that we can see, do we see the same effects that we see in mice in humans? And so in phase 1, the first question we answered is Ezutromid well tolerated and suitable for future testing? And we found that yes, indeed it is. So now we're in the phase 2 trial where we get to ask, can Ezutromid modulate Eutrophin in patients with DMD? And can it have a positive effect on biomarkers of muscle structure and health? Now in the future, what we want to ask is, does this have positive effects on muscle function? And does it have positive effects on the quality of life for patients? Phaseout DMD is an open label trial. It's 48 weeks in length. We've had 40 patients total enrolled, both in the US and in the UK. And in this trial, what we discovered, so we had a data monitoring committee that looked at the safety of the trial so far, and they decided that this trial was safe to continue so that we could extend the trial so that the boys can be on Ezutromid until the drug is either discontinued or it's approved. And we also are including this safety arm. So that's to allow patients that have been in previous phase 1 trials but were not eligible for this phase 2 trial so that they can continue to receive Ezutromid. And we also have a different population to look at safety with Ezutromid, which is important to us as we develop this. So we'll really be looking at 3 buckets of information in this trial. We're looking at a whole host of endpoints. This trial is really aimed at trying to answer, can we have any effect on the DMD disease in patients? So we'll first be looking at the mechanism, and so this will be done through biopsies. And we'll be looking at the uniform, looking at the Eutrophin distribution. We'll also be looking at muscle fiber regeneration in those biopsies. With muscle health, we'll be looking at the fat fraction from the MRI, and this is actually our primary endpoint. And finally, we'll be looking at functional measures. So this is your 6-minute walk distance, the North Star Ambulatory Assessment, and the performance of upper limbs. Now, I will caution you that this trial isn't set to be optimized to see functional differences, but we are measuring them just to check that out. So we're really focused on the mechanism and the muscle health. And so what we want to see here is for the Eutrophin, we want to start to see that more uniform distribution. So that's much like the honeycomb-like appearance that we saw in dystrophin in a non-DMD muscle. We'll also be looking at fiber regeneration, and we want to see a decrease here. Now, it sounds a little counterintuitive. We'd think we'd want boys to be making more muscle, but what we know from the biology of Eutrophin is that when it's present, it's usually present when muscles are regenerating. So we want to see a change so that we have Eutrophin present and it's in mature muscles. And so we want to see the decrease in muscle fiber regeneration. And then with the fat accumulation, we want to see a slowing or a stabilization of that fat fraction as measured by MRI. Now, if we get positive results in Phaseout DMD, we'll next want to run a larger global pivotal trial that will likely be placebo-controlled. And data from that trial, we think, could support accelerated or conditional approval. And part of that approval means that you have to run a confirmatory trial after that. Now, if you want to get more information from us, you can sign up on eutrophintrials.com to get periodic emails from me that gives updates on our Eutrophin modulator program. And you can also find out more information about the trial on the clinicaltrials.gov. And finally, I'm around, so you can always ask me questions if you don't get your question answered now. But here's my email and phone number if you have any questions after that.